Hi ladies and gentlemen, today we're going to extract DNA from an organism. So a food item would be great, anything that is mushable. So I have chosen strawberries here. These were frozen, but any food item that is raw and non-processed is going to do well, that you can mash up. If you need to use a blender to do it, that's fine. First, we're going to go over all of the ingredients and materials that we need. We need a food item that's mashable. We need a plastic baggie that seals. For step 2, we're going to need some sort of jar that you can cap so you can shake up. You need some salt and just some regular dish soap. The next step, you will need a funnel. I did not have a funnel in my house, so I found a plastic bottle that kind of got smaller at the top, and I cut off the bottom. Now I have a funnel. Next, you will need something to be able to strain it with. This is just a laundry bag. I use them to get my fruits and veggies at the grocery store, so I don't have to use plastic. But you can really use any material that is going to allow the liquid to go through. And then you need some thin, tall, clear item that you're going to put the DNA in, right? But you're going to want it to be thinner and taller as much as you can. This is an old spice jar that I didn't have full anymore. Lastly, you're going to need some really cold ethanol or isopropyl alcohol. You can get it from the pharmacy, isopropyl alcohol or rubbing alcohol. Ethanol is really good if you can get that. If you can't find any of those items, vodka can work really well in a pinch, but it's going to have to all be very, very cold. So please keep it in the freezer overnight first. You also will need either a little eyedropper or a pipette, but if you don't have those items, I understand. Make sure that you have just a pen and another little baggie, and I'll show you how to do that in a little bit. Last item is you're going to need some sort of like wooden skewer to be able to pull your DNA out, and that is it on the materials. Step 1. We're going to have to mash the strawberries really well, or whatever food item you chose. So let's look over here. And the better you mash it, the better it's going to be in getting all of our DNA out. So make sure you get all those little pieces. Step 2, we're going to make the extraction buffer. So let's take our jar, let's add some salt. We don't need to make measurements off of this. Just make sure you have a lot in there. Add some dish soap. My dish soap is pink, so it matches my strawberries, but it's best to use dish soap that is as clear as possible. Now I'm going to add some water. Try to add warm water to it. Cap it. Shake it all up until it's completely mixed. Step number 3, we're going to add the extraction buffer to the mashed strawberries and mix it really well, because we have a whole bunch of soap in here and it's going to break up the phospholipid bilayers. You don't need to add too much. Now, reseal it and thoroughly mix it, and make sure that you have everything mashed and broken up. Step number 4, we're going to separate out the big pieces from the rest of it. It should hopefully have our DNA in it. So let's come on over here. We have our funnel, We have our material that is going to separate it. And then I have my thin, tall glass to catch it. Make sure that you don't pour in so much that it goes too high. You want it to be about halfway full. Oops. It is looking really good. Step 5. Now we're going to make the DNA insoluble in the alcohol and it will rise up. So you need your really cold ethanol and a dropper if you have one, but if you don't have one, it's going to be okay. I'm going to show you a trick. Have your bag. Take your alcohol, pour some into your bag. Seal the bag completely. Turn it upside down. You're going to poke a hole in just the corner here. Now you have a nice little eyedropper. So let's come on over here. And you just want to squeeze it down the sides. The key here is to be really patient. We want the ethanol to layer out on top. You can already see a clear layer forming on top. Okay, notice you have all of these strands rising up and it's starting to collect on top. The DNA is going to be really attracted to itself. And so, finally, you can grab your skewer, your wooden skewer, and start to collect it. That is all DNA, ladies and gentlemen. And the more that you play around with it, the more it's going to stick all to itself. See? Congratulations. I hope you have fun. Have a great day.